Students, today we are going to see about what is backcross. So backcross is a cross of F1 hybrid with any one of the parental genotypes. The backcross is of two types. They are dominant backcross and recessive backcross. So here we can see F1 hybrid is crossed with any one of the parent is called as backcross. So any one of the parent means it may be tall plants or dwarf plants. Okay. So any one of the parental genotypes means, so heterozygous, that is F1 generations, it is crossed with a dominant character, that is dominant parents, and F1 generations of heterozygous tall plant, it is crossed with a recessive parents. So backcross is a cross, that is F1 hybrid is crossed with any one of the parents. It may be tall or homozygous tall or heterozygous tall. Okay. It involves the cross between F1 offsprings with either of the two parents. Okay. When the F1 offsprings are crossed with the dominant parents, all the F2 develop dominant characters. No recessive individuals are obtained in the progeny. So when the F1 offsprings, that is heterozygous tall, it is crossed with the dominant parents. All the F2 generations, all the F2 develops what characters? Dominant characters. There is no recessive characters. No recessive individuals are obtained in the progeny, and all were what means? Tall plants. No recessive is present. All were tall plants. Capital T, capital T, capital T, capital T, all were tall plants. Okay. So this we say, that is when the F1 offsprings are crossed with the dominant parent, all the F2 develops dominant character. No recessive individuals are obtained in the progeny. Same like that, when the F2 hybrids are crossed with a recessive parent, individuals of both the phenotypes are appearing equal proportions, and this cross is specified as test cross. Now we can see what is test cross. Same, the F1 offsprings are crossed with a recessive parent. So recessive parent, the individuals of the phenotypes are appearing in equal proportion means we can see tall 2, and dwarf 2. This we call as equal proportion. So even the F1 offspring is crossed with the recessive parent, and this the proportion is 2:1. So this we specify it is a test cross. Okay. So the recessive backcross helps to identify what means heterozygosity. So this we call as heterozygosity. What is present in the F1 generation? It is crossed with the dominant character. F1 generation is crossed with the recessive parent. So this character which is present at present we formed with the F1 generation heterozygous tall, that is it is heterozygosity. So we can see the recessive backcross helps to identify the heterozygosity of the hybrid. Okay, so this we call as backcross. To understanding, I draw this checkerboard. And next one is, we are going to see about what is a dihybrid cross. So dihybrid cross means the crossing of two plants differing in two pairs of contrasting traits. We have to cross the two plants with the different characters. Okay, contrasting, two pairs of contrasting traits means two pairs of contrasting characters is called dihybrid cross. So Mendel considers that seed shape, that is round and wrinkled, and cotyledon shape yellow and wrinkled as two characters. Seed shape, that is round and wrinkled shape, and cotyledon yellow and green. In seed shape, round R is dominant over the wrinkled shape R. So we can see this is the seed shape, that is capital R. It is dominant over green, that is a wrinkled green. And next one is cotyledon color yellow capital Y is dominant over green Y. So cotyledon color capital Y and what? Cotyledon yellow color is dominant over green color cotyledon small y. Okay. Here the pure breeding round yellow parent is represented as by the phenotypes RRYY, capital RRYY, and the pure breeding green wrinkled parents is represented by the genotype small RRYY. So this is a pure breeding plants. Homozygous round seeds, yellow cotyledons, homozygous wrinkled seeds, green cotyledon, with the parent capital R, YY, and small r, small y, small I. So this is a pure breeding plants. So during gamete formations, the paired genes of the characters assort out independently of the other pair. So genes are located on the chromosomes, that is a different chromosomes, assort independently during meiosis. Many possible combinations of gametes has been formed. Okay. So this we called as otherwise segregation of gametes. There is no dependent upon the gametes. No dependent upon the gametes. The gametes which is present it is assort independently during meiosis. Okay, the resultant gametes thus be genetically different, and they are of what following 4 types. So after F1 generation, there is a 4 types of gametes has been formed. So capital R, capital Y, and then capital R, small y, smaller capital Y, smaller small y. So this is the 4 types of gametes which is present in F1 generations. It is independently assort during meiosis. So we can see 4 types of gametes has been formed during F2 generations. Yellow round with the capital Y and capital R, 9 by 16. So in the checkerboard, that is Punnett square, we can see 16 boxes. So 16 characters of plants has been produced. Out of that, yellow round is 9, yellow wrinkled is 3, green round is 3, and green wrinkle is 1 by 16. Okay, so these are the 4 gametes. This gametes has been formed, 4 gametes. This 4 gametes is kept in the right side, that is male gametes, and this side, that is a female gametes. Okay, so this 4 gametes has been right and 4 gametes has been kept in left. Okay, when you cross this, capital R is dominant. Always we have to put the capital R first and then the recessive one. Capital R, capital Y. Capital R first, capital R, capital Y, small y, and this to this. Capital R, small r, capital Y, and this capital R, small r, capital Y, small y, and come to this second line. Capital R, capital Y, small y, and come to this capital R, small y, capital R, small r, capital Y, small y, capital R, small y, and third to this, and fourth to this. Okay, so from this we can see 9:3:3:1 with 4 types of characters has been formed. Okay, by crossing the 2 plants differing 2 pairs of contrasting characters. So what is the ratio obtained in a dihybrid cross means 9:3:3:1. Okay, so these 4 types of gametes in F1 hybrids unite randomly in the process of fertilization and produce 16 types of individuals. Okay, 16 types of individuals in F2 in the ratio of 9:3:3:1. So this is what dihybrid cross. So dihybrid cross follows the rules of law of independent assortment. One more question. So law of independent assortment means, so you see what is law of independent assortment. So capital R, capital Y, and small r, small y. So the gametes has been present for this gamete F1 hybrid. 4 types of gametes has been formed. These 4 types of gametes are present in separate chromosomes. Independently segregated, not during meiosis, independently segregated, not dependent on other gametes. Okay, not dependent, independently segregated means the law of independent assortment. So this rules followed with the dihybrid cross. Okay, so what is the ratio of dihybrid cross means 9:3:3:1. So what is the ratio of a dihybrid test cross? So another experiment followed by Mendel is, if the F1 hybrid, that is round yellow piece, okay, F1 hybrid la kadicha and the round yellow, capital R, smaller, capital Y, small y, is crossed with the homozygous double recessive genotype, smaller. You know, this F1 hybrid is crossed with any one of the parent. It's called as a test cross. We already seen that. If the F1 hybrid is crossed with the F1 hybrid which is formed during dihybrid is crossed with the recessive parent means that is a wrinkled green, and green, that is smaller, small y, small I. So what is the ratio obtained means the 1:1:1:1, all in the same proportion, that is 25% of each characters, and this we call as dihybrid test cross. So what is again I am telling, when the F1 hybrid is crossed with the homozygous recessive genotypes, the ratio obtained is 1:1:1:1. Okay, so we can see these 4 types of gametes is present in the male gametes, and this RRYY in the left side. So when you cross this, we can see round yellow and round green, wrinkled yellow and wrinkled green in the ratio, genotypic ratio, and then phenotypic ratio means external appearance of the parents, and what is the phenotypic ratio for 100%? Each one carries 25 and 25. Okay, so this we call as dihybrid cross. So now we see about what is back cross and what is called dihybrid cross. The ratio of dihybrid cross means 9:3:3:1, and follows rules of law of independent assortment. And last one, dihybrid test cross, F1 hybrid cross with the homozygous recessive parent is called dihybrid test cross, and ratio of 1:1:1:1. So read this topic, 3 topics clearly, and follow the test. Thank you.